Thanks for the kind introduction and the invitation for the organizing committee of Open Box Science. I'm Alan. I'm a postdoc fellow in Humboldt Lowell's lab at BWH, Harvard Medical School. So today I want to share to you a story about LAND-Vs. So just a quick overview before we get lost. So LAND-Vs are a new structure. So first of all, I'll talk to you about what these structures are. And then I'll talk to you about one of the functions and the mechanism of function of these LAND-Vs that we discovered. Thirdly, we'll talk about LAND-Vs in physiological conditions like endogenously. And finally, I'll touch upon the biogenesis. And then one more little secret treat for you guys. So let's begin. So in our lab and myself, I've been working on neutrophils for more than 10 years. So neutrophils in the human circulation, about 40 to 60% of our white blood cells are neutrophils. They're the most abundant white blood cells. So many of you have seen this video from David Rogers in 1953 about a neutrophil chasing a bacteria. So they're very highly effector cells. However, they have a really short half-life. So just allow me to give you an introduction on the neutrophil half-life. So neutrophils are produced from stem cells in the bone marrow in the process called granulopoiesis, where then they exit the bone marrow and enter our circulation. And if all is good, they undergo cell aging and death, and everything is fine. However, if there is an infection in a certain tissue, let's say a lung, kidney, or liver, they can transmigrate and chemotax via cues or even swarming through into the inflammatory tissue and perform a plethora of functions, which include forming neutrophil extracellular traps where they release their DNA. They have direct antimicrobial effects through oxidization and also peptides. They can directly engulf and kill the pathogens. And then people usually believe after they've also all is good said and done, the pathogen is cleared, they are erythrocytosed by macrophages, or they reverse migrate back into the bloodstream and then go back to the liver or the bone marrow to be cleared. So the dilemma is their half-life is only 18.5 hours. We make so many of them. So actually each human on average makes about 10 billion of these cells per day just to have them die. And normally, hopefully most of the time we're not sick. So is there maybe an underlying function to this evolutionary phenomenon? It's not only humans, mice, zebrafish, neutrophils all have a very short half-life. So the fundamental question is, do they need to die to prevent something? Or does their death serve a function? Or maybe both. So that's basically the central question that we're trying to answer. So this is just a time-lapse video of neutrophil death. It's stained with an Exon 5 PI, and then you can see that as time goes by, more and more cells die, so they become PI positive. But as you can appreciate, the morphology, it's not everything is uniform, first of all, and not everything dies at the same time. Some are just small green cells, some become these puffed red cells that are classified over here on the right. So that means the neutrophil death is a heterogeneous and complex process, which many people agree and work on. But then again, about the function of them. So it was actually during one of these video sessions that I discovered that as time goes by, these neutrophils, which in here, they form these really large vesicles that bud from them. And to make such a large vesicle would require significant amounts of energy and actually actin cytoskeleton rearrangement. So we sought out to maybe look at what are these things and do they actually function? Do they do anything? So based on their size, we did a very simple sequential centrifugation protocol just at 3000 G to pull them down. And then we looked at our purification, so looked at WGA, negative stain TEM, and also cryo-EM to sort of characterize their size to be about 1 micron. So it's larger than your typical exosomal microvesicles. Also, and then thus we term these vesicles large aging neutrophil-derived vesicles, LAND-Vs for short. These LAND-Vs actually were more enriched during later times of neutrophil culture. So as you can see, the blue line is where how many neutrophils are left. So LAND-Vs are actually enriched at the 24 to 48 hour time point, meaning at the later times of the neutrophil life, that's where these guys are produced. LAND-Vs themselves actually are pretty stable. So once you isolate them, they can actually be stable in vitro culture for about out to 5 days, whereas neutrophils die off pretty rapidly. So what do these LAND-Vs do? We tested their functions alone, like do they produce ROS, do they move around? No, nothing of that. So thus, because they're formed from neutrophils, so we wanted to see if they have any function in the neutrophilic inflammatory system. So we used a acute pneumonia model where we used S. aureus as our target bacteria, injecting these bacteria directly into the lung. At 1 hour, we would administer either PBS or 50 micrograms of LAND-Vs directly into the lung. And then we collect the BALF and lungs at 24 hours to assess inflammation process. Again, if you have any questions, you can interrupt me or the host can moderate this. Thank you. So at 24 hours, we found that when you adopt a transfer LAND-Vs, you have significantly less white blood cell infiltration, less neutrophil infiltration, suggesting less inflammation. This coincided with lower levels of IL-6, interleukin-1 and interleukin-6 pro-inflammatory cytokines, also suggesting lower inflammation. Histological-wise, we also saw that compared to the PBS-treated group, the LAND-V-treated group had significantly less inflammatory lungs, so you can see that the lung tissue is less swollen and also less neutrophil infiltration. This also coincided with a less protein amount, showing less edema and less vascular leakage, suggesting alleviated lung damage. And when using a lethal dose of S. aureus, 1 dose of LAND-Vs could actually improve the survival. So the red line is when you just challenge with the lethal dose. And then this black line, the square black line, is when you give LAND-Vs. It has a improved survival, also lower clinical score. Interestingly, though, while neutrophil numbers and also pro-inflammatory cytokines were decreased, the bacterial load and also the macrophage numbers were not, suggesting that LAND-Vs could modulate inflammation without compromising immunity. So just a little background on extracellular vesicles. So currently, the field is a little bit messy, but currently I think the easiest way to accept this is this sort of classifying extracellular vesicles into exosomes and ectosomes. Exosomes are preformed inside the cell, either way, shape, or form, and then this packet of vesicles would fuse with the cellular membrane and then be released. Ectosomes, on the other hand, in again, way, shape, or form, would use a budding system, either apoptotic blebs, microvesicles, or other larger or more specialized ectosomes. But basically, everything is actin cytoskeleton mediated and budding from the cells. So clearly, LAND-Vs would fall into the ectosome classification. So thus, we wanted to see if this anti-inflammatory effect were specific to LAND-Vs, or maybe all neutrophil vesicles per se could do this. So thus, we wanted to compare first with apoptotic bodies, because neutrophils are short-lived. They do form apoptotic bodies. These apoptotic bodies usually are, the role of apoptotic bodies is just to keep everything quiet, get efferocytosed by macrophages, cause IL-10, and thus being anti-inflammatory, and because of their phosphorylserine on their surface. So we also compared that with the liposomes, phosphorylserine liposomes as well. So again, giving the mice equal amounts of LAND-Vs, liposomes, or apoptotic bodies, we found that actually only LAND-Vs could induce this anti-inflammatory phenotype, shown here in neutrophil numbers and also histology. We then also wanted to compare if this anti-inflammatory effect could be seen with neutrophil exosomes and microvesicles. And again, these microvesicles and exosomes, as you can see with the neutrophil recruitment and also the histology, could not rescue this phenotype, suggesting that this is a LAND-V specific phenotype. So how are these LAND-Vs special? Because a cell can make various types of vesicles. So we wanted to tease this apart to show that it's not produced just during death, but also aging. So previously in the lab, we also found a cocktail we named it Clone G, which is basically a mix of inhibitors, which we can prolong the neutrophil lifespan up to 5 days. So as you can see here, compared to the black line, Clone G in red prolong neutrophil lifespan. And actually looking at LAND-V production, because we prolonged its lifespan at later times, whereas you can see the increase of LAND-V production instead of decreasing, suggesting that LAND-Vs were produced as a function of neutrophil aging, but not neutrophil death, because when you have more death, as in the black untreated group, you don't have more LAND-Vs. The LAND-Vs are produced from both untreated neutrophils and clone G treated neutrophils. Both could have this anti-inflammatory effect, suggesting that it's not clone G that's changing the properties, but rather it's a function of aging and, again, independent of cell death. Thus, that leads us to this simple model, whereas a neutrophil ages, it forms LAND-Vs, and these LAND-Vs are distinct from the apoptotic bodies and the apoptotic cell itself from a dying neutrophil. So these are a novel type of vesicles. So what are LAND-Vs? What would LAND-Vs entail? So we took these vesicles, we performed a spectral flow. I showed you 20 colors here. We did also a 40 color panel, and then did unbiased clustering to see what do they compose of. So we found that this cluster 2 in orange here had this enrichment of CD55 and CD47, which I'll get to in a bit. And it was the most abundant group in the LAND-V populations. This is just a high resolution, super resolution confocal to show this vesicle that it indeed has C66B, which is a neutrophil marker, along with CD47 and 55. Also doing mass specs to compare the proteomes of LAND-Vs and its neutrophils paired, we also found that the LAND-Vs were enriched in proteins involved in the complement system and also coagulation pathway. And lo and behold, among the complement system proteins, CD55 was there again. So finally, to validate this via Western blot, we show that LAND-Vs were enriched in CD55, whereas other neutrophil proteins such as myeloperoxidase, lactoferrin, metalloprotease, and neutrophil elastase were all decreased. So to see if this anti-inflammatory effect was mediated by CD55, we in vitrally blocked CD55 or IgG, and then incubated put these LAND-Vs into our pneumonia model to see if it could still retain its anti-inflammatory effect. So obviously, when you put LAND-Vs alone, you have this anti-inflammatory effect. However, when you pre-treat with anti-CD55, but not with IgG, you lose this anti-inflammatory effect, suggesting that this anti-inflammatory effect we saw was CD55 mediated. We also did similar studies with using knockout mouse LAND-Vs, so CD55 straight knockout, and we also sorted the LAND-Vs CD55 positive and negative, and found that only the CD55 positive LAND-Vs could have this effect. So just a little bit background on what CD55 is. But before then, let's introduce the complement system. So the complement system is a very, very ancient innate immune system comprising of 3 main pathways. Now, recently there was a Nature paper that gave a 4th pathway, but the classical, lectin, and alternative pathway. Whereas the classical and lectin pathways usually are more pathogen-driven, whereas the alternative pathway is a spontaneous activation of the complement, something called tick-over, and then it would all converge to a C3 convertase, 2 different forms, C4C2 and C3BBB. So C3 is basically and then C3 could C3 can be further cleaved into C3A, which can serve inflammatory functions. C3B can opsonize pathogens or microbes, and also form the C5 convertase to further have effector functions. So as you can see from this diagram, the C3 is the central hub of the complement activation. Thus, usually, the way to control it in our bodies, we usually just shut down the availability of C3, or we inhibit the amplification. So CD55 is one such protein that inhibits the C3 convertase formations by kicking away BB. So just to make sure that if this phenotype was CD55 mediated, as we've shown, but also through complement. So we looked at the same mice that we showed in the previous slide and found that indeed, when you treated LAND-Vs, the C3b opsonization complexes were decreased and the increase of the availability of the substrate, which is the C3 alpha chain. And when you block CD55 on the LAND-Vs, you restore this to the PBS levels, suggesting that CD55 on LAND-Vs do work through complement. To further show that, we isolated LAND-Vs directly from the BALF, and we stained it via LY6G to identify neutrophil-derived vesicles. And then we did an in vitro binding to recombinant C3b and showed that these LAND-Vs bound to C3b in a CD55 dependent manner. So when you add anti-CD55, you block the binding. And finally, to confirm this LAND-V CD55 complement axis, we use heat-killed S. aureus bacteria because in the C3 knockout mice, if you give it a live S. aureus bacteria, the mice would die immediately. So we gave it a heat-killed S. aureus and assessed its inflammation in wild-type or C3 knockout mice. So as you can see here, again, in the wild-type mice, when you give LAND-Vs, you have significantly decreased neutrophil numbers. But in the C3 knockout mice, you lose this effect, suggesting that LAND-Vs works through C3. So if you don't have C3, there is nothing to inhibit. To further prove that, we actually supplemented an exogenous C3. So we gave the mice extra C3, and we can revert the LAND-V effect, suggesting that indeed it's the C3 that's the major player here. So thus, this is our working model. As neutrophils age, they produce LAND-Vs. And on the surface of the LAND-V, usually if there's no CD55, the convertase forms C3bVb. It further amplifies complement. And as if LAND-Vs have CD55, it binds to the C3b, kicking away the BB, and thus stopping this amplification and stopping this pathology. So how does this happen in vivo? Because we've been giving the mice LAND-Vs, but what about normally? So because there's no reliable way to just knock out LAND-Vs, and we're still working on it, specifically. So the best thing we can do is just knock out CD55 on neutrophils specifically. So we built this mice, a CD55 tissue-specific knockout versus its flox litter mates, and then we infected it with the S. aureus and did a time course. So as you can see through this plot here, the C3 complex is much more enriched if you knock out CD55, suggesting that if you lose CD55 on neutrophils, the complement activation in the lungs throughout this time course is much more severe than the wild type. And this was also shown here by neutrophil number recruited, so you have much more abundant neutrophil recruitment and much severe pathology. Finally, and then also we found that this binding of C3b, as we showed, is LAND-V specific. So when you knock out CD55 on the neutrophils, the LAND-V forms obviously don't have CD55 and cannot bind to C3b either. Finally, to definitively show that these CD55 LAND-Vs are the major players in modulating this immune response, we infected a round of wild-type or knockout mice. We sorted the LAND-Vs from their mnemonic BALF, so as you see here, the LY6G positive. We put these endogenous LAND-Vs back into either wild-type or knockout mice. So let's start with the wild-type first. So in the wild-type mice, you give wild-type LAND-Vs, it works. And there's anti-inflammatory effect. You give it a knockout LAND-V, there's nothing happening. In a tissue-specific knockout, you have increased pathology as shown in the previous slide. However, if you give the wild-type LAND-Vs, the endogenous wild-type LAND-Vs, you actually can restore that to a lower level. And obviously, if you give it a knockout LAND-V, you have no effect, suggesting that it's the LAND-Vs that are playing the role in this immune modulation. So moving on, can we actually see LAND-Vs during inflammation? Or does this actually happen? So these are human samples. We got donors from human lungs, healthy and pneumonic. And then we stained and quantified, so you can see the green is C66B, which is a neutrophil marker. And I'm just enlarging for you. You can see that in the healthy lung, you have a little bit, just maybe one out of the whole field. But however, in the pneumonic lung, you have many of these DAPI negative, but C66 positive. So traditionally, pathologists have always brushed this off to say, oh, this is like antibody staining or nonspecific binding. But we show that it's actually more than that, because we also built a mouse model which expresses GFP on neutrophils and orange on every other cell, and we induced it with pneumonia. So this is the lung section of it. And as you can see, there's many of these DAPI negative vesicles that are just in the alveolar space and also in the parenchyma space, suggesting that LAND-Vs do exist and it's not like just an antibody staining artifact. So, but how do LAND-Vs stay there that long? So we actually wanted to see like the physiological role of LAND-Vs. So this is and we know, since we know that it works through complement, the complement activation curve during this pneumonia, it starts at 2 hours, peaks at 6 hours, persists until 24, and drops off. Same thing with the C3b complexes, suggesting that it's acute at 24 hours. The neutrophil number also matches this, peaks at 24 and rapidly drops out at 48. However, the LAND-V numbers in vivo all peak at 48 hours and persist to 72 before dropping off at 96, suggesting that LAND-Vs actually are enriched when neutrophils are already gone. So LAND-Vs, how do they stay there that long? So again, this is an exon-5 staining of a LAND-V produced. You can see that it glows green brightly, suggesting that they are phosphostyrene positive, so they should be eaten. So we wanted to test this and see how it can stay that long. Looking at it, we found that there's 3 main don't eat me signals on LAND-V, 24, 31, and 47, and 24 being the most dominant one. We next did an in vitro J774 macrophage efferocytosis assay where we labeled the LAND-Vs and then just incubated them with macrophage to see how much they take up, and compared that with antibody blocking. So normally you have a set amount of LAND-Vs taken up. After you block CD24, you have much more increase inside the J774 macrophages, suggesting that CD24 is the dominant don't eat me signal for mice LAND-Vs. And if you triple block, you have the same effect. So we test this again in our pneumonia model where we blocked don't eat me signals on the LAND-Vs and then subjected them and see if we can recover it. So at 24 hours, we used the CD45.1 LAND-V and put it into CD45.2 mice. We found that normally you can recover about 13%, but once you triple block or just block CD24, you have a much lower recovery rate. And this much lower recovery rate suggests that it's gone, taken up, or destroyed. And this decrease of recovered LAND-Vs also coincided with lower levels of anti-inflammatory effects, suggesting that these don't eat me signals help these LAND-Vs stay there for a longer time. So again, this is our model on LAND-Vs. Besides CD55, you have the CD24 don't eat me signal to prevent macrophages from being eaten, whereas other vesicles were just taken up by macrophages. And finally, with the final few minutes, I want to talk about how LAND-Vs are formed. So CD55 usually expressed on neutrophils. On a fresh neutrophil, it's all around the cell, normal. However, though, when a neutrophil ages, CD55 becomes polarized on the cell, and then LAND-Vs form from this polarized site. So thus our model is a fresh neutrophil. Through aging, it drives this polarization, and then the LAND-Vs form from it. This is just imaging flow data showing the same thing. You have full length CD55 and it's unpolarized and polarized neutrophil, and sometimes you see these enriched CD55 buddings from it. And the fraction of this polarization, CD55 polarization, increases over time. And this MFI on the neutrophils would decrease over time because it's giving away its CD55 to the LAND-Vs. This is not specific because CXCR4, on the other hand, increases over time. So this decrease is not just not specific. So again, to show this, we see that this is live imaging with CD55 staining. You can see that the CD55 polarization happens, it clusters, and then there's a vesicle bubble forming from it, suggesting that this polarization, clustering, and LAND-V forming actually happens. CD55 polarization. CD55 is a GPI-linked protein. It's usually associated with lipid rafts. We wanted to see if it indeed this polarization happens via lipid raft clustering, and through IF staining, indeed it does. Furthermore, we also wanted to see if CD55 actually coincided with ordered lipids. So because lipid rafts have been shown to have more ordering of lipids, and you can see this is a polarized neutrophil with CD55 polarized. The red line and green line, the red line, the polarized section has a higher ordered lipid. And thus we know that when a neutrophil is aging, it's more polarized, and thus the GP value, which is a function of lipid ordering, also increases. And finally, looking at the polarized site, you can see that it has a significantly higher polarized value than that unpolarized site. And finally, we wanted to just look at if we can disrupt lipid rafts and then we can disrupt LAND-V formation. So indeed, when we treat with MBC, or AAY9944, these two disruptors of cholesterol, we saw a decrease in CD55-LAND-V counts, and the CD55 MFI on the neutrophils retained, whereas on the LAND-V is decreased. So to finally bring us to our model is when the neutrophil age, the CD55 polarizes where LAND-V buds from it onto the ordered lipid raft part, whereas the unordered part has less ordered lipids and less CD55. So finally, in lieu of the next presentation, Peter, who is my good friend, I wanted to share this data, which is we took a cohort of COVID patients, and then looking at their plasma, we actually found, we could find LAND-Vs in the human plasma, and we found that actually the LAND-V count in the plasma negatively correlated with disease severity. So that means that in patients that have milder COVID, retrospectively, of course, they had more LAND-Vs in their circulation. So because every human being is different, so could this LAND-V count sort of chicken and egg problem contribute to this milder disease? We don't know yet. Interestingly, though, the number of LAND-Vs actually negatively correlated with the number of neutrophils in circulation. So it wasn't just more neutrophils, more LAND-Vs. It actually was opposite, suggesting that it was a better resolution. So finally, just to conclude today's presentation, during pneumonia, neutrophils are recruited to the infection site where there's a lot of bacteria. They serve their function. And in this inflamed tissue, in this case the lung, the complement system is also activated to perform a plethora of functions. As neutrophils age, usually the CD55 is used for self-protection. But as it ages, it polarizes on the lipid rafts to release LAND-Vs. And then this LAND-V would then inadvertently talk to the complement system and inhibit it. So putting this into a broader context, basically neutrophils have a bunch of functions, but most of them, even through death. So for instance, we know NETs, they contribute to autoimmunity, vascular occlusion, all these interesting things. And also their lytic death through pyroptosis or other forms of lytic cell death can release a lot of DAMPs and cause pro-inflammatory. Currently, their only anti-inflammatory response is a passive one, which is just getting eaten up, taken up, and having macrophages do all the heavy lifting. But today, I presented a way that through neutrophil aging, they can actively form these vesicles and talk to another arm of the innate immune system. And via CD55, they can actually inhibit and basically tone down inflammation. So this is the first report that this active function actually happens with neutrophils. So with that, I want to conclude my talk. I really want to highlight a few people. Hongbo Luo, my PI, who was really supportive of this project. Qinxiang Huang, a great collaborator who helped me with a lot of the work. Hao Wu's group at BCH, who helped me with the cryo-EM and the characterization of these vesicles. And also Jay's group at BCH, who helped me with a lot of the biophysics. So with that, I thank you, and I'll take any questions. Appreciate it.
Congrats, that was an excellent talk and very impressive results.